As I mentioned earlier, we had a lot of really outstanding abstracts submitted for our poster session this year. And so we decided, instead of selecting just one session of short talks, to select 2 sessions and to focus the second session on some of the ways that the stem cell community has stepped up to fight the COVID-19 pandemic in so many different ways. So we're very proud to spotlight these talks in a special session, and they will also be featured at the poster session later on for further discussion. So our first speaker is Kristina Dobrindt, who's a postdoc in Kristen Brennan's lab at Mount Sinai here in New York, and she'll be speaking about the impact of genetics on infection by SARS-CoV-2. Kristina.
Thank you so much for giving me the opportunity to speak. I hope you can all hear me clearly. So today I want to tell you about common genetic variants and why they're important, not only for SARS-CoV-2 infection. And sorry. Okay, so as you all know, SARS-CoV-19 is extremely variable. The main symptoms are pulmonary, but there are also neurological and gastrointestinal symptoms, with the severity ranging from asymptomatic to severe and extremely critical. And the main players are the ACE2 receptor, TMPRSS2, FURIN, and CD147, where TMPRSS2 and FURIN cleave the spike protein gene to enable infection of SARS-CoV-2 into cells. And I want to tell you a bit more about common genetic variants now. What we understand about common genetic are single nucleotide polymorphism, SNPs, so single variants in the genome that are present in at least 1% of the population. And our typical genome has 3.5 to 4 million of those SNPs, but most of them are inherited together as blocks in so-called linkage disequilibriums. And we study these with genome-wide association studies by connecting a certain trait to certain SNPs in a population. And the result we get are Manhattan plots, where you can see the chromosomes on the x-axis, and then the association with the trait and the significance on the y-axis. Each line you see here is one risk loci, and each dot you can see is one SNP that is associated with risk. So everything above the red line is a significant threshold. This is the Manhattan plot of schizophrenia, which we're usually working on. But by now we also have Manhattan plots in vivo studies for COVID. On the left-hand side, you see hospitalized COVID versus the population. And you see very few extremely significant loci, whereas on the right-hand side, you can see self-predicted or self-reported COVID versus non-COVID. We have way more risk loci with a little bit less significant. And looking at our host genes, we can see that all of these have risk loci in the cis-regulatory region. We are especially interested in FURIN because we've actually already been working on FURIN. So FURIN has a schizophrenia risk allele. It's RS4702, which is extremely common. It's present in 34 to 45% of all alleles, and it is the mostly associated risk SNP in its risk loci, and can clearly be connected with the risk with fine mapping. And what you also have to know is that 50% of all SNPs are expression quantitative trait loci. This means they regulate the expression of a transcript. In this case, the risk allele GG leads to a downregulation of FURIN. So you can see it down here in postmortem brain. We have a iPS cell model for this. By editing this allele in iPS cells, you can generate isogenic lines that only differ in this one SNP, generating AA neurons and GG neurons. And the risk neurons with GG have indeed a lower FURIN expression, which is, by the way, mediated by MIR338, which also leads to functional effects, for example, reduced neurite growth. So our question we had is, does this risk SNP also influence SARS-CoV-2 infection? And which host genes are actually important in which cell types, and does it make a difference? The setup we have are starting with pluripotent stem cells, differentiated them in NGN2 neurons, alveolospheres, intestinal organoids, and knock down our host genes of interest and analyze with high content imaging and qPCR. First question is, how relevant is furin in brain, and can we infect brain cells? So on RNA-seq data, you can see that CD147 and furin are highly expressed compared to ACE and temperas, which can also be confirmed on Western blot. And also in pre- and post-infected neurons, you can see that the furin expression is robust compared to other host genes. And we can infect neurons already at a low MOI after 24 hours and can get a robust interferon response. And also on ICC level, we can see some infected neurons, and on Western blot level, robust replication of SARS-CoV-2 in neurons after 24 hours with a high MOI. But we wanted to know if this is only an in vitro artifact or if you can also see an effect in vivo, looking at hamster brain. Again, see high furin expression compared to some of the other host genes, and a vast up and down regulation of transcripts, and a very early interferon response, which dies down again after a few days. Longer lasting are the transcriptional effects with upregulated transcripts being involved in membrane trafficking and abnormal cell migration and Golgi vesicle transport, and downregulated genes, even more interestingly, involved in postsynaptic membrane and neurotransmitter activity, so directly influencing the function of our brain. Our other system I'm going to present here are the alveolospheres generated from iPS cells. We have a robust expression of host genes, can infect the alveolar cells on ICC and on qPCR level. We're interested in is how the different transcripts, how important the different transcripts are. We studied this by down-regulating our host genes with short hairpins, and we could see that ACE plays the most important role in alveolar cells, followed by CD147, furin, and temperas. And even clearer on high content imaging, ACE has the most important effect, and but also furin plays a role. Neuron, it looks a little bit different. Again, even though ACE is only lowly expressed, it plays an important role, followed by furin. So furin has definitely an important function in SARS-CoV-2 expression. Now we're looking at our SNP variant. Here, the whole alveolospheres, and on top, the AA, the wild-type AA SNP, on the bottom, the risk GG SNP. You can already see that we have a higher infection in our AA alveolospheres. Can confirm this on two different isogenic pairs. On qPCR level, we have higher furin in the AA compared to the GG, and also much higher infection. You can appreciate that there is already in these two genomic backgrounds a lot of variability between infection of genomic backgrounds, which is quite interesting. And on neuronal level, we also have a striking difference in furin expression depending on the genotype, and also here we see a difference in infection. So we can prove that, or show that we have one common SNP located in the 3' UTR of the protease furin, and which impacts alveolar neuron infection of SARS-CoV-2 in vitro. And this has truly been a team effort. I want to thank my supervisor, Kristina Dobrindt, and the whole team, the Icahn lab, the Huckins lab, the Hanover lab, and everybody else. Okay.
Thank you, Kristina, for presenting that work. That's really important to look into the genetic basis of this susceptibility. I was curious, do you have any phenotypes that you're looking at besides infectivity in terms of just sort of the impact of the virus on these different cell types that you've examined?
So we're currently waiting for RNA-seq data to come back to look at these phenotypes as well, and to see a wider transcriptional modification due to infection.
Right. And are you planning to look at some of the other variants as well, like the ones in ACE2 that you had described earlier?
We're not looking at ACE2, but we're currently looking at top hits from the recently published COVID study to see how they're impacting the infection.
Yeah, I was going to ask about that study. Okay. Great. Excellent.